Sorry about that. Yeah. As I was saying, most of the existing approaches, predictive models, only focus on disease versus healthy, but there are more complicated uh, uh, outputs like you have in terms of brain diseases like Alzheimer's, schizophrenia. You have neuropathological features like cognitive impairment, cognitive diagnosis. You have track scores which talk about neurofibrillary tangles which are associated, which are not basically final outputs or final outcome like disease was healthy, but they are all intermediate phenotype that we want to predict. Uh, the third one is biological interpretation, which a lot of us and lot of us have been focusing on. Is even though we use a lot of these machine learning and deep learning algorithms, what we want to do is once we have the algorithm ready or the model ready, we want to go back and look into it and identify what is happening in the model, why is it predicting this, and what features are useful and helpful. Um, there are some key challenges to these motivations that I talked about. One is heterogeneity of multi-omics data. For example, you have let's take a case of gene expression, which is a continuous variable, whereas genotype is like 0, 1, and 2, which talks about zygosity. When you look at imaging, the heterogeneity comes in terms of whether the imaging features that you take are from the same region as your gene expression data, or from the same tissue, or from the same cell line, etc. The second one is biological interpretation. As I said, if you take a complicated deep learning model or a machine learning model, it is very hard to interpret the biology behind it. And finally, the missing modalities is a huge issue due to several factors, like you have experimental bottlenecks, sample availability, etc. It is very hard to fetch complete set of multiomics data. Uh, and oftentimes only a partial, you have only a partial availability of this data for different individuals. Even then, I mean, there are a lot of cases where you have full availability of these multi-omics data, but the amount of the number of samples that has complete data is very limited. So there are a couple of existing approaches that does phenotype prediction from genotype or other related data. Uh, the, it can be categorized into three different groups. The first group is the association studies, like you have GWAS, EQTLs, and TWAS, all of those studies. But the key drawback of that is association is not always causation, and they don't predict; they just associate. The second one is you have machine learning approaches, like you have popular polygenetic score, predict scan, which they first take genotype and then try to impute gene expression, and from gene expression they try to they try to come up with they try to predict the trait or a phenotype. So the problem with these approaches is that they treat each individual feature or each individual omics individually. They don't combine multi-omics or multi-feature together. And finally, you have multi-modal integration, like you have various, in the recent years advancement, there are various number of approaches that does multi-modal integration. But there are two different drawbacks. One is black box nature of these machine learning approaches. The second one is they consider just the disease outcome as phenotype, which per se is not a huge drawback, but it can be treated as a drawback. Revisiting our goal was to use multiomics data to build a machine learning algorithm to integrate this data and then do a phenotypic prediction and also do biological interpretation. That is where our model DeepGAMI comes into the picture. Uh, so as you see here, our model has we taken this is an example, but I'll show you cases where we take different modality approaches. Over here, we have gene expression and genotype as the input as two different modalities. And what we do is in the first layer, we call it biological drop connect layer. In that layer, we introduce biology into the model itself. Where, like for example, we have gene regulatory networks, and we have EQTLs. So we introduce those into the model itself. So we want to let the model know that we are guiding you through this. You learn the weights, on your own, but this is what you have, this is where you have to go towards. And the second part of it is the auxiliary learning layer, where we estimate one modality from the other. We don't estimate the exact modality itself, we estimate the latent space of each modality so that we can do cross modality imputation. And finally, we have a feed-forward neural network layer, which does phenotype prediction. That is on the top panel. When you look at the bottom panel, so the top panel is during the training phase. In the bottom panel during the testing phase, since as we're using the auxiliary learning layer, it enables us to only input one single modality as an input to the model, and we can estimate the other modality latent space and use that to make the phenotype prediction. So as I said, biological drop connect layer is where most of us have heard of dropout layers, where the dropout layer drops out a particular node per se. In drop connection layer, what it does is you have an edge between two different nodes. The nodes are dropped randomly. What we do here is, instead of that random dropping of the nodes, we say that you drop if there is no link between a gene, between a transcription factor and a target gene, which is a TRN, or if there is no link in terms of EQTLs between a SNP and a target gene. So that is what we introduce as biological drop connection layer. The second one is auxiliary learning layer. It is a very straightforward linear transformation. We use the main task is to estimate latent space representation of one modality from the other. We have two different parameters, alpha and beta. We just try to estimate that. And we use mean square error loss between the original latent space versus the estimated. So this enables us to do cross-modality imputation. The way we train is we split the data into 80% and 20% for training and testing. During the training phase, we do 5-fold cross-validation and and also to train different hyperparameter tuning. Once we have the best model from the optimal set of hyperparameters, we use it on the test set to get the results. We use balanced accuracy because most of the times, the data that we have is highly imbalanced. So balanced accuracy is one measure that can account for this. And we also use AAC scores as the performance metrics. The last function that we use, minimize, consists of two things. One is primary loss, and second is auxiliary loss. The primary loss is the loss between your prediction versus the ground truth. The auxiliary loss is the mean square error that I just talked about for the auxiliary learning layer. The next step is we want to interpret. We want to prioritize genes, SNPs, or the links between the transcription factors and the target genes, etc. So what we do is we use something called integrated gradient approach, which what it does is, based on your model and your input, it tries to estimate which of the features are useful, which of the links are useful. It gives you a score based on which you can further go ahead and do downstream analysis to see these genes are useful, or these you can prioritize genes, SNPs, or the regulatory sub networks within a regulatory networks, etc. These are the data. So we applied our model on 3 different data sets. One is schizophrenia versus control. We wanted to start with the very basic, phenotype, which is disease versus healthy. Then we went to RoseMap data, which contains Alzheimer's disease, different phenotypes, like you have cognitive diagnosis, Brack score and Sierra score, which talks about mild cognitive impairment, no cognitive impairment, all of those. And finally, we also applied it on the mouse data set that we had for brain, where this is a more complex phenotype where you have 6 different mouse layers and we wanted to predict those. So in terms of the schizophrenia data set, we can see that on the for the schizophrenia data set, we had 2 things. One is on the bulk data from the TLPFC brain region. And the other thing is we collaborated with another lab from Mount Sinai, where they had developed an algorithm called BMind, which was able to impute cell type gene expression data, right? So we use that. So the results that I'm showing you here is for both bulk and cell type level predictions. On the top left that you see is the AU curve, and we can clearly see that uh, the blue and the red one is our model. We can clearly see that both dual modality, where you key in both the modality as input and you key in only single modality as input, our model performs very good. Similarly for each different cell types. And on the bottom, what we have is this is the prioritized gene regulatory network and EQTLs. We can see that we have certain set of cell type specific schizophrenia related genes, like, you have FOSL2 in microglia, and then you have REV3L. These are genes that are schizophrenia associated, but also cell type specific. Next, we applied it on Alzheimer's disease. Over here, we have on the top left, you see we have three different phenotypes. The first one is the CRED score, which says AD probable versus Alzheimer's definite versus no AD, and we have BRAC score, which is a binary classification, early versus late stage. And finally, cognitive diagnosis, which says you have a cognitive impairment, or no cognitive impairment, or mild. So we can see that our model performs five-fold cross-validation, gives us better accuracy, even if we just input one single modality as well. The middle one here is the independent cross-validation, where we have the 20% held out test set. Over there also, we have fairly decent performance on it. What we then did was we did the prioritization of the SNPs and genes, and we gave it to a pathway analysis. And we can see that there are, I'm just marking one or two of them, but we can see that there are a couple of AD related pathways that come up from our prioritized genes and SNPs. And finally, we applied it on the mouse visual cortex layer, which has, so that over here, the data set is different. It's not gene expression and genotype. Here, we have gene expression, and we have something called electrophysiological features, which are the responses to certain set of stimulus in mouse brain regions. So we have gene expression, electrophysiological features. We predict different layers in the mouse brain region. There are a couple of existing machine learning approaches that does predict this mouse visual cortex layer. The bottom part of it shows the different approaches, and ours is green and red, which we see that outperforms existing approaches. And on the right side, we see that, so on the right, what we have is, we had certain set of mouse cells which were only available for layer 4. So what we did was we only applied, and it did not have electrophysical features, it only had gene expression data. So what we did was we wanted to see how it performs, and we can see that it performs very good. In conclusion, I present you interpretable deep learning framework, which is used for improving phenotype prediction. And we introduce a novel auxiliary learning layer, which does cross-modal imputation. And finally, this model enables you to do phenotype prediction, not just disease was healthy, but complex phenotype prediction, even when some modalities are missing. Our future work is to extend this to have more modalities like clinical data or imaging, and we just have 2 modalities. Now we want to also extend it to having 3 or 4 different modalities as well. And we see that a linear transformation in the auxiliary learning layer performs well, but we want to also go ahead and look at nonlinear transformation so that we can estimate cross-modality better. Our preprint is available here, so we are waiting. We have submitted it to briefings, and we are waiting for the response. Ours is an open source software, and it is available on GitHub. Anybody can use it. with this, I thank a lot of there are a lot of people to thank and acknowledge. One is the entire lab and the people who have helped me and in dealing with working with this project. I thank lab from Mount Sinai and from University of Pittsburgh, which helped us fetch the data and give us insights and also give us cell type specific data. And that's it. I can thank you. If you have any questions, I'm willing to take.
Thank you for a great talk. My question is about the drop connect layer that you have. Have you compared the performance of your model when instead of using a biological network that connects your TFs to genes, you connect them through a randomly generated network with the same in degree and out degree of the nodes and see how the performance changes? The reason I'm asking is because in another application where we looked at in that comparison, we could get very comparative performance when we generated random networks, which led us to believe that the effect that it has is more of a regularization effect on the network.
Yes, we did try out the random drop connections where we dropped randomly, and then the performance was very bad. We had like a 50%
with the same degree, with the same in degree and out degree similar to the original model.
I did not catch
the degree distribution. We're matching the original.
Yeah.
Okay, very good. Thank you.